All right, I give it another go. Is this better now?
Can you try again, Nils?
Hello, hello, hello.
Yeah, I will say it's still far, but so I would like you to maybe loud a little bit your voice. Yeah, I think should be fine.
Yes.
Excellent.
Yes. Okay, thank you. Because I just checked it with my colleague also via WebEx, and this worked fine. So maybe it's the connection. I don't know.
Yeah, now it's fine. Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuh. And very good afternoon, very good morning to all of you. It is a great honor for me to welcome all of you to this webinar organized by the Department of Biology, Faculty of Science and Mathematics, Sultan Idris Education University, in collaboration with the Malaysian Proteomics Society, MAPS. I would like to extend my greatest appreciation to our distinguished speakers, Professor Dr. Bernard Kuster, Professor Dr. Sebastian Carpentier, and Dr. Nels Rosen for their kind willingness to deliver a speech for today's webinar. Congratulations to the organizer and all parties involved for making this event possible. Ladies and gentlemen, let's all listen to a short opening recorded message from Associate Professor Dr. Muhammad Faisal Nizam Lee bin Abdullah, the Dean of the Faculty of Science and Mathematics, Sultan Idris Education University.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. And a very good afternoon to all of you. It is a great pleasure to welcome all of you to this webinar, Updates on Proteomics Research, the Arabidopsis Proteome Atlas and Crop Proteomics Anno 2022, from blind high throughput shotgun approaches towards single-cell proteomics, organized by the Department of Biology, Faculty of Science and Mathematics, University Pendidikan Sultan Idris, or in English, Sultan Idris Education University. Alhamdulillah, I would like to extend my greatest appreciation to all speakers, Professor Dr. Bernhard Kuster, Dr. Nils Rosen, and Professor Dr. Sebastian Carpentier, to the organizing committee and delegates for your contribution that made this webinar a success. Indeed, it is a great pleasure to be a part of this significant event. Congratulations to the organizers and all parties involved for making the event possible. Ladies and gentlemen, this webinar aims to provide a platform for proteomics researchers to exchange novel ideas and establish cooperation in research and innovation. I strongly believe that this is a very good opportunity for all participants to extend their knowledge to various areas of research and innovation. In addition, the high-quality research to be presented in this webinar will enhance the culture of research and knowledge interchange. I hope that this event has been a good platform for all participants in inspiring and exploring a new frontier of proteomic research in the future. Ladies and gentlemen, hence, I would like to use this webinar as a stepping stone to invite you proteomic and molecular biology researchers to any research and innovation collaboration at University Pendidikan Sultan Idris. Through this collaboration, it is hoped that we will come up with high quality collaborative research and move forward to improve the knowledge of proteomic researchers. With Bismillahirrahmanirrahim, I declare the webinar Updates on Proteomics Research officially started. Thank you. Wabillahi taufiq wal hidayah. Assalamualaikum warahmatullahi wabarakatuh.
Special thanks to Associate Professor Dr. Muhammad Faisal Nizam Lee bin Abdullah for the opening speech. Ladies and gentlemen, we will have 3 speeches for today's webinar. followed by question and answer session at the end of the webinar. This is the plan for today's program, so please stay tuned and get ready with your questions. You also may leave them in the chat box, and then the respected speakers will try to address them at the end of the session. I also want to announce that the webinar will be streamed on YouTube, and the link will be posted in the chat box. It is a privilege for us to have Prof. Bernard Koster, one of the pioneers in the proteomic research field in the world, to be with us today. Prof. Bernard will give his talk together with Dr. Nels Rogen. Prof. Bernard Koster is a chemist by training. He obtained his PhD in biochemistry from the University of Oxford. He did a postdoc funded by EMBO long-term fellowship at the EMBL in Heidelberg and the University of Southern Denmark in Odense. After 7 years as Vice President of Analytical Sciences and Informatics at the biotech firm Silzum, now known as GSK, he became full professor of proteomics at the Technical University of Munich, TUM, since 2007. Prof. Bernard's research focused on 3 main topics. The first one is mapping proteomes to identify which genes exist as proteins, where they are expressed and in what quantities, how they are post-translationally modified, what their lifetime and further properties of interest for understanding biological systems. The second focus is to investigate how therapeutic cancer drugs work at the molecular level to understand their often multiple mode of action. And the third, to build proteome tools, particularly in the areas of sample preparation, liquid chromatography, MSMS, and informatics to improve the technological-based proteomic technology. Prof. Bernard has received an ERC Advanced Grant in 2019, aiming to integrate proteomics into precision medicine. He's also a member of the German National Academy of Sciences. He received a number of awards, notably the Discovery in Proteomic Sciences Award of the Human Proteome Organization, HUPO, for mapping out a draft of the human proteome. He has published more than 200 papers on proteomics and bioinformatics, and has co-founded 2 startup companies that operate in the area of proteomics and artificial intelligence. Ladies and gentlemen, let's all welcome Prof. Bernard Koster. Prof. Bernard, the mic is yours.
Yeah, thank you very much for this very kind introduction. I'm sharing my screen now in the hope that everyone can see the top slide. Jamil, is that working? Okay, very good. Yeah, thanks again for your kind introductory words, and good afternoon to Malaysia, good morning in Germany. So I'm very happy to give this speech today, although I would much rather be in Malaysia myself. I haven't been, so at some point I should come and visit. I'm very happy also to present to you today because this is the beginning of a collaboration between a couple of labs between Germany and Malaysia, which is always a great thing in science, that we can operate across borders much more easily than some political constraints often allow. So science goes across many parts of the world, all across the world. And what I'm trying to tell you a little bit about today is something that we've done about a year ago to create a draft of an Arabidopsis Proteome. And before I start this, I should make a few disclosures. Jamil has mentioned a few of them. I founded in 2 biotech companies. We get some research funding from industry and also in-kind support. None of this really has any influence on how we do our science, but for good practice or transparency, I'll just tell you about it. Also, in the introduction, it already featured what my lab is doing in general. We're looking at recording what is in proteomes and try to understand how drugs work in building proteome tools. And today, I'm going to venture out in the plant sciences field, which is a bit of an extraordinary thing for me to do because most of my research actually happens in mammalian systems. But nevertheless, it's a fascinating world that we've entered with my lab. And so I'll tell you a little bit about what we have done in trying to map out the Arabidopsis proteome. And as was mentioned also in the introduction, is the main motivation seemed to be fairly simple, but I think it's fundamentally important to understand, to find out or to understand which proteins are actually expressed as a protein from a genome. Even though many genomes have been annotated quite well, just something that looks on the nucleotide level like a gene because it has all the right elements doesn't necessarily mean it's going to be expressed as a protein. So we think there's a fundamental interest in just mapping out which proteins exist. And then, of course, a little more interesting perhaps is where within the plant or the organism are these proteins expressed and in which rough quantities. And there are a couple of surprises that one finds even with these very simple questions. And of course, we would want to understand how the expression levels are controlled and how they are post-translationally modified, because the latter in particular is one way how nature has been able to create functional diversity from a rather limited set of genes. And so here on the topic of Arabidopsis, the picture on the right-hand side just means to illustrate which parts of that model plant we've looked at to create parallel proteomes, transcriptomes, and phosphoproteomes from that, and that is shown here. And one nice thing about working with plants, it's usually not a big problem to get good enough amounts of starting materials. So we mapped the proteomes of all of these tissues, starting from about 1 milligram of total protein per tissue. Getting 1 milligram of protein from pollen is, of course, much harder than from a leaf, but it can be done. And what you see on this rather complicated chart here is, starting from the very same tissue material, we split up the samples into two ways. One for going down the RNA sequencing track to generate transcript levels, and then to the left-hand side to go through the protein track and split that up into a protein expression proteome and phosphorylated proteome, and use mass spectrometry and chromatography to analyze or to capture the data, and then some informatics down the road. So, and just to give you an idea of what the effort was like, we spent about 2 days for each tissue to generate a proteome, and a couple of hours for the phosphoproteome. So it is quite an effort, but it's doable. And then importantly, the RNA-seq material data was collected from the very same sample, so that we hope to be able to have good comparative analysis also on that end. A couple of numbers. So after the end of quite a bit of work, here's just a data summary. On average, we got about 13,000 transcripts per tissue and about 12,000 proteins, and about 9,000 phosphorylation sites per tissue. And all of that data is actually already available in Proteomics DB. This is an informatics project that we've had going for a long time, together also with the software company SAP, in which we basically have put the human proteomes, the Arabidopsis, the mouse is in there, although it's not yet published, and a couple of other proteomes. But perhaps what's more important also is to, or more interesting, is to take a look at a comparison of these 3 proteomes that were essentially created by the same technology, and to look at how many annotated genes there are. And this is not really new, that humans have about 20,000 genes, the mouse has 10% more, and Arabidopsis, according to our report, has quite a few more. And then annotated isoforms, it's quite a different story. And that doesn't mean to say that Arabidopsis has the least isoforms. It's, I think, just less well annotated. So that probably number is probably going to go up. But now when we look at the current state of play, what proteomics can do, it looks as if we can capture about 70% of the genes that are annotated as protein coding genes. So we are by far not complete. We're missing about 30%. And then the question, of course, arises, are these 30% actually really proteins, or are they expressed at so low levels that current technology can't get them, or are they expressed under circumstances that we haven't captured in just by looking at normal physiological conditions? Because that is what we've done. And the answer to that is a bit of everything. And it, of course, means that we are not really at the end of this, but this is going to go on until we can claim to have seen the majority of any proteome. effect. Okay, but delving in now into the Arabidopsis, both proteome, phosphoproteome, and transcriptome, this is what the data looks like across all of these tissues that we looked at. And the color code is just basically grouping all of these tissues into families. So everything that's green is obviously the tissues that have green color around them, and in brown it's seeds and embryos and so more the developmental tissues. And what is quite interesting to see is when we look across the proteomes of all of these tissues, about half of those proteins we find actually everywhere, even though in every tissue we get about in excess of 10,000 proteins, only half of those we find actually in every tissue. On the phosphorylation side level, that's even lower, even though we can capture about 10,000 phosphopeptides in each experiment. The ones that are really shared in all of them is really small, and that alone already says that there's a lot of regulatory diversity even within the same organism across different tissues. And to compare that then to the transcript level, same story. Also on the transcript level, the number of transcripts that are shared across all of those tissues is actually rather small. So there's a very great diversity of gene expression, both on the RNA as well as the protein level, and even more diversity on the phosphorylation level. And in order to try and sort of get some order into this group in Stockholm has proposed a scheme where you basically say what is actually specific or not specific, or what is globally expressed. And one of the really surprising findings is that genes that are really expressed in a tissue-specific manner, meaning that they are only expressed in a single tissue, this is a very small proportion, both on the transcript level and on the protein level. So one has to kind of rethink the textbooks a little bit when we teach our students that protein expression is highly cell type specific. I think we need to qualify this because on the whole, a lot of proteins and things are expressed in many places, not in all, but in very many, but maybe just in very different quantities. So it's more a matter of quantity than a yes or no answer. Okay, and now a couple of further sort of higher level views on these three levels of omics. When we look at what is reported in ARA report 11, it's about 27,000 genes on the transcript level. When we add everything together across all of those tissues, we actually near completeness on the transcript level. It's about 25,000 transcripts. When we put it all together on the protein level, not quite as much, 18,000, and the phosphoproteins, that's also interesting, but in line with what we see for humans. And about half of all proteins are phosphorylated at least once. And in total, then this makes up for more than 40,000 sites. So there's a lot of phosphorylation also happening in Arabidopsis, not really new news because it actually has a lot of kinases. But what we keep thinking is interesting to look at is also the dynamic range of expression. So how much of a gene is there, or how little of a gene is there? And when we look at the transcript level and you use as a quantitative measure some normalized count level, then essentially what we can see is a distribution of transcript abundances here in gray that for the most part span about 3 orders of magnitude in abundance. And when we map onto that, the proteins that we found, we can see that it actually covers the higher abundance part very well, and then at the very low abundance part, not quite so well. But it's interesting also that even though there are transcripts that express at very high levels, we can never find the protein. And part of the reason is biochemical. We can maybe still not extract some of those proteins efficiently from mitochondria, where there are a lot of mRNAs made from mitochondrial proteins. But there may also be just very high levels of the transcripts, even though they never really turn into a protein. Now, what is interesting then to compare and contrast to the proteome level, when we do the same plot, but now use an intensity-based absolute quantification measure on the protein level, we can see something that is quite different in terms of the distribution. While the mRNA levels are roughly distributed across 3 orders of magnitude in abundance, for the proteome, or a maximum of 4, for the proteome that is more than 6 orders of magnitude. So that alone also says, for technical reasons, it is much, much harder to cover a proteome completely compared to transcript level, because the abundance distribution in the proteome is so much higher. And then when we map onto that blue distribution here, those proteins for which we have also found some phosphorylation evidence, you can actually see that phosphorylation evidence follows exactly the abundance distribution of all proteins. So essentially, it says, technologically speaking, we are probably not biasing our phosphoproteomes any longer towards the high abundant proteins, but actually we cover the whole range, which is actually quite good in terms of the ongoing discussions, how much in terms of technological variation there is in proteomic data. In fact, when you take a really close look at, usually the proteomic data these days is more robust than the transcriptomic data. Okay, and when we look at across a couple of interesting protein classes, we can still see like kinases, phosphatases, transcription factors, or transcriptional regulators, and ask how much have we actually seen. It's still fair to say that the transcript actually sees most of them, and the proteome sees about 3/4 of them, except for the transcription factors, that many of those are still not covered. Either they are super low abundant, or they are being very rapidly degraded after they've been made. That is also a possibility. So the transcript is there, but the protein may not be there. And then we also, one of the particular reasons why we even actually do this is, as I said, we're trying to understand which annotated genes actually exist as proteins. And in turn, if you turn this around, you can also confirm or not whether an annotated gene actually is a protein or codes for a protein. So, and that's what we try to do here. And UniProt has come up with a scheme that assesses the evidence for proteins. And basically, it's a simple scaling from 1 to 5. And evidence level 1 in UniProt would mean there is actually experimental evidence at the protein level. Somebody has found that protein by whatever method. The evidence level 2 is that a transcript for that gene has been found. So at least one knows it's being transcribed, and therefore perhaps it is also translated into a protein. But evidence level 2 doesn't mean that, it means that people haven't seen a protein so far. Or evidence level 3, that protein is just inferred from the fact that the gene that codes for it is homologous to another gene, perhaps in another organism, for which it also codes for a protein. So that's an informatics evidence level. And number 4 is an even lower evidence level where one just predicts that this is a protein coding gene. And then there's evidence level 5, whether there is entirely uncertain whether this thing is a gene that will ever code for a protein. So, and when we look at now at our own data for this and ask that question, how many proteins are annotated in UniProt with what evidence level, and how many of those did we actually find? We actually do find the vast majority of the ones that UniProt already lists as protein evidence level 1. We actually see a very, very high number also of proteins for which only transcript level information was available. And then you can see the bars go down. But in total, our Arabidopsis Proteome provided actual protein level evidence for about 12,000 genes for which no protein had been at least not been annotated in UniProt before. And we think that's a major value of the work, so that we can actually know that these transcripts actually not only are transcribed from genes that look like protein coding, they are actually protein coding. We often take this for granted, but I think it's important still to show that this is actually the case. And it's also good for the transcriptomic people, because they can now be much more confident that the transcripts that are being measured on RNA-seq level will actually this phosphatase here, or this U-box protein here, or this sulfotransferase up here, so that the cell is obviously able to make a certain amount of protein from an mRNA, but for this protein, it actually makes from the same quantity of mRNA, it makes 10 times more protein. Or for this particular gene, it makes 100 times more protein. So there's clearly a regulation that we can see in these plots, that some mRNAs are much more efficiently translated than others. And obviously that would be, and I don't show it here, but this does correlate also with the total abundance of the protein, in the sense that it makes a lot of sense for very highly expressed proteins to be also efficiently translated from a given number of transcripts, because that's one way of for the cell to be efficient. If they had to make one-to-one amounts of mRNA, they would have to make a lot of mRNAs. So it's much more efficient to be able to accommodate many more ribosomes on a single RNA, if the cell knows that it has to make a lot of that protein. So, but then what is interesting, so even though at the same transcript level, we can have vastly different amounts of protein, but when now we look for each protein, and these are those 3 proteins here now across all tissues, and ask the question, what is that ratio of protein and transcript levels? Then we actually see for many, many proteins, particularly if they are abundant, this is practically the same. No matter where in which tissue we look, for this, so for transferase 18 here, that median protein to transcript ratio is about 21. So why is that interesting? So once you have established this for many, many proteins, you can use, in fact, transcript data, RNA-seq data, to have a good guess as to how much protein there will be. And that also seems that then there is a strong sort of code in the mRNA that basically knows how much protein to make. So it's not entirely constant, but it's relatively constant here. It's relatively constant here. It's a little more shaky here, but this is also on the level for which there is just less protein. Okay, and then we found actually something that surprised me more than I had expected. When we now look at the proteome expression characteristics of individual tissues, which is here shown group by tissue, and the protein densities as a fraction of the total of individual proteins. So you could actually, you can see that for each of those tissues, often one single protein dominates the expression of this. So of course, in the green tissues, it's Robisco. Of course, people would have expected this. However, I keep forgetting the numbers, but I think in the literature it says something that leaves also the 30% of the protein content, let's say in leaves, is Robisco. According to our data, that's actually not the case. It's just about 4 or 5%. So you see, it is the most abundant proteins in leaves, sure, but it's in the literature has been vastly overestimated how much of the total this Robisco actually is. And then surprisingly, let's say in the seed tissues, it's actually a single protein that dominates by far all proteome expression, which is a storage protein, which of course makes sense. But then this is in relative much more high abundant than even Robisco in leaves. So that surprised me that that this could be so extreme. But interestingly, then when we only look at, let's say, the top 100 proteins that I'm jumping to the right-hand side here, when we only look at the top 100 proteins in each tissue, we actually find that only few, usually and sometimes half of them, are shared between tissues. So basically that says again that a single protein or a handful of proteins can be extremely abundant in one tissue, but not very abundant in other tissues. But what is interesting about it, maybe not so much from in the plant world, but we can actually, just by looking at the 100 most abundant proteins, or we can actually tell you without ever knowing what sample it was, we can tell you whether it was a leaf, or whether it was a seed, or whether it was a flower, or whether it was a pollen. We would have never even had to know that this is what it is. The protein expression pattern would tell us this. And in a way that when we now go to the middle, we can actually, this is the principal component analysis of all these proteomes, and one can roughly see the green tissues kind of are together, and some other families of tissues are together. But pollen is always, no matter what we do, pollen is always very different. And obviously that's an interesting tissue, of course, to look at when it comes to how plants, of course, replicate. Now that was on a global level, but you can also break that down to families of proteins if you're interested in that. And here, for example, we have a couple of cases of a family of kinases. And basically what you see is, again, across all of those tissues, we plot here the relative proportion of the different members of the AGC kinase family. And you can see that in each tissue, it looks a little different, but again, in pollen, it is very, very, very different. So this yellow kinase AGC 1.7 is very, very highly abundant in this one, but not so much in others. And that one can actually also then do by staining, can follow up whether that is the case. So again, what that says is there is tissue specificity in proteome expression. Of course, that's what people might expect, but the extent to which this happens is often quite surprising. And again, the interesting or the good news for the community is all of this data is out there. And so whoever is interested in a particular protein can look this up quite easily now. And this is more of the same in a sense now, but looking at protein complexes. And in particular here, the cotamere complex, when we look across all tissues, that's just the abundance of the cotamere complex in different tissues, and it varies a bit, but not too much. But now we can also break this down into the different subunits, and you can see that this by and large is quite similar. But when we look at now, zoom in particular into the proportions that certain subunits have in different tissues, we can actually see that particularly for these subunits here, there is a very strong difference between those embryo and seed tissues compared to the rest. So that the makeup of the composition of that complex for this particular subunit actually varies a lot between the tissues, where it, for this complex component, it really is the same everywhere one looks. Or here the relative proportions of this one, of this one, of this one are also not so different to each other, but these components obviously are extremely tissue specifically, and not only expressed, but also then assembled into protein complexes. Now, also a word on the phosphorylation level. You can do similar things. You can ask the question, how much phosphorylation is there in which of the tissues? And you can group that by the families of protein kinases. And again, the pollen stands out here. It has a lot of signaling capacity. The same you could do for the phosphatases, same story here. So it stands out also in its ability to signal. And then you can, of course, also start to look for phosphorylation motifs in trying to address or begin to address which kinase might phosphorylate which site. And here's just a couple of these motifs that we found, that there are motifs that are followed by a P or a basic residue, or something that has on the C-terminal side various acidic residues, and or also on the N-terminal side a couple of basic residues. And that implies that, of course, there is specificity in these kinases. And we hope that our data is going to help the people also to make more connections of this type in the future. And then just one more word about phosphorylation is shown on this plot here on the left-hand side, where also the levels of phosphorylation, we often immediately relate phosphorylation with signaling, and that it is super low abundant. But then there are many more functions that phosphorylation may have. And actually what we've plotted here is for all proteins in Arabidopsis, we just plotted the content of serine, threonine, or tyrosine as part of all of the amino acids. So it ranges roughly from 10% to, in some cases, up to 45%. But we plot that then against what the proportion of serines or threonines or tyrosines that are actually phosphorylated. And then these LIA proteins really stand out, where almost 90% of all of their serines and threonines and tyrosines are actually phosphorylated. And here's more examples of those. So a lot of proteins are very, very heavily phosphorylated, and they are constitutively phosphorylated. So they're phosphorylated once and then it never comes off. And our interpretation of this is that phosphorylation of these proteins that are so highly phosphorylated may have something to do with creating compartments in a cell that don't have a membrane around it, but are characterized by phase transitions, and phosphorylation would be one way of accomplishing this. So with this, I think at the end of my little overview of Arabidopsis Proteome, and there's a lot of people in my lab who have helped doing this, I should point out in particular Julia Merkner, who's the first author on this paper, who's left the lab by now, but she has done most of the actual experimental work and data analysis. And of course, I'm thankful for the funding that we received to doing the research. And one could now ask the question, where do we actually go from here? And that is what Niels is going to talk about in a moment. So I don't know if you want to take questions now or if we leave them to after Niels' talk. I leave that to the chairperson to decide. Thank you very much.
Thanks a lot, Prof. I think we proceed with Dr. Nell's speech, and then after that we can take some questions maybe before Prof. Sebastian's speech start.
Okay.
Sorry, you only saw the presenter mode, right? Sorry, 1 second. Okay, I guess you see the presentation by now, right?
Yes.
Fantastic. All right. Okay, then also hello from my side. So Bernard just already started talking about this. So we were, after this huge Arabidopsis project, we were thinking what could or should actually come next. And we actually thought, let's now take a closer look at actually, the proteomes that feed the world, right? So really to now go into actually the crop plant world, so that we now really, leave the model plant Arabidopsis and really go into the actually plants and crops that become food at the end of it. And the impact or the idea behind this is I think quite obvious. Of course, plants are the very basis for all life on this planet, and especially for our daily nutrition. And it's still the same question. Proteins in the end control more or less all molecular aspects that happen within a cell, within an organism, and actually learned quite a lot about model plants like Arabidopsis, and now also about its proteome and especially its genome. We have very little knowledge about crops, and this is rather true for all crops on this whole planet. And to change this, we actually formed an international network with more than 30 or 40 partners by now from around the world that want to help us to change this. So the initial idea of this project is to collect the 100 most important food crops from this planet and map their proteome in a depth that is comparable to what we did with the Arabidopsis proteome. And what is especially interesting today for many of you maybe is that we actually funded a new PhD program that I will talk about later a little bit, where we have the chance to get up to 18 different PhD students starting today. So if you have talented students or interested students that could imagine to work and live for a few years in Germany, it would mean a lot to us if you could spread the word around this. But I will come to this later again. First of all, so how did we decide what the 100 most important crops on this planet are? We actually used data from the FAO. So I guess you all know that they have a huge database system called Faustat, which is very rich in information about the crops from around the world. And we actually used the global production in millions of tons of the year 2018 to rank all crops listed by the
sorry, Nels, I'm so sorry for the interrupting, but can you go a little bit louder if you do not mind?
Yes, is this better?
Yeah, as loud as you can. Yeah.
Yeah.
All right.
Okay. I'm trying to shout. Sorry, sometimes from this.
I'm so sorry for that. All right. Thank you.
I just repeat this slide. Okay.
Yeah, please go ahead.
Okay. So the question is, how do we decide what crops are part of this project and what not? And we actually used statistical data from the FAO that lists the global production in millions of tons of the year 2018. And by this, we made a ranking. And then this comes to no surprise that, of course, at the beginning of this ranking, you have the global players, sugarcane, maize, or rice. And at the lower end, you find upcoming stars like lupins. So I expect lupins, for example, to be in the middle 50s after the next 10 years. And at the very low end, you find, for example, still very important crops like vanilla orchids. And so what we basically did is we counted from 1 to 100 and said, these are the 100 crops that we want to work on. So how do we now map these proteomes? We actually, just like in the Arabidopsis project, we aim to collect 10 to 30 different tissues of each and every crop, extract the proteins with some help of, for example, a tissue lyser. And we always try to get roughly only 200 micrograms out of each and every tissue. And then we have a workflow that is comparable to what we did in the Arabidopsis project. So we also, we digest the proteins, we fractionate the peptides. And the only difference this time is that we actually now have a pipetting system, a robot that actually does all this work for us so that it's easier for us to do, that it's feasible to do, and also, of course, that the reproducibility is way higher than what we did actually last time. And so we aim for 6 different peptide fractions per tissue, and then everything is transferred to microflow as the MS analysis. And then, just as Bernard already told you, we actually have a nice set of different softwares that really helps us to get as much information out of all these huge data that we create at the end. And yeah, it's basically the same question, but just this time for way more organisms. So we're still interested in which genes are actually expressed as proteins and where can we find them, and also in what quantity. So this is basically just the same questions that we had for the Arabidopsis project. And our aim here is to go for more than 10,000 proteins in each of the 10 to 30 tissues or organs. And we actually played around with quite a lot of different workflows to see if this is possible. And I'm happy to say that we actually found a way to get these ID rates. So here again, you see the 6 fractions that we now make with our robots. And you can see that we can actually identify up to nearly 100,000 peptides. So these are Arabidopsis data. So we use Arabidopsis to get the workflows running. And you can also see that with help of a ProSet software, we can actually really reach this goal of more than 10,000 proteins per sample. And we do not only want to map an organism, we also have some, well, classical biological questions that we want to work on this time as well. So for example, we got very interested in mapping the fruit development of tomatoes. So from the fruit set over the expansion up to the ripening process. So from little small green to up to the big red tasty ones. We really want to map what's happening within the proteome during this development. And you can, of course, then find new dormancy factors, because we actually know already quite a lot about fruit development, especially in tomato, but there are still many things that we do not fully understand. And we could actually also think about some rather funny ideas, like, for example, finding the proteome part that is responsible for making this tasty at the end of the day. And we get even more depth in this experiment, because we actually dissect each and every step of this development into subtissues. So we have the seeds, we have the locular tissues, we have the placenta, and we do this for each and every stage. So in the end, we actually have a quite large sample set for the fruit development of tomato alone. So only for one part of one of the 100 crops. And getting all this material is actually only possible because of this great partner network that we established. So this network is growing and growing. So we have, of course, many partners here at the Technical University of Munich, but we also have found great partners in Malaysia. We have the EMBL. We also have many German universities and breeding institutes, but we also have partners from China, Japan. We have Africa, Peru. We have many members of the AOAPO. And yeah, as I just told you, we also have found 2 great partners from Malaysia. So we have Jamil, who is helping us with date palm samples and also jojoba. And we also have CF from the University of Nottingham, Malaysia, who actually tries to help us with durian, lila orchids, sweet potatoes, and winged beans. And the thing is, we are not done yet. So you can imagine that if you want to collect all these 100 different crops, you need a lot of partners. And we're actually still looking for partners. So if you actually have crops that you work on and that might be important and useful for a project, it would mean a lot to us if you would actually contact us, and we would then try to organize a video chat with you and see if we can come together here. The other thing actually that we do not just need the crops, we also need manpower. And we were actually lucky to get a new PhD program funded. It's an international PhD program, and the idea is really to recruit the best people from around the globe and get them all to us in Bavaria and to work with us on crop proteomics and to really bring crop proteomics into the next generation of what is possible. And yeah, as it says here, we actually have, I think, currently money for 12 to 16 PhDs. And I want to point this out again. We're really looking for international people. We do not want 18 German PhD students. We want people from around the globe. So if you have interested PhD students, please let them know or let me know. And fewer words on what this program is actually about and how we organize it. We have this thing running that we call the crop proteome engine. So all the plants are processed here in our labs. So we do the protein extraction, also a lot of the greenhousing, the mass spectrometry, but also the whole bioinformatics part. And I think you could just see from Bernard's talk that bioinformatics is actually key to really get the interesting information out of these huge data sets that we create. And we actually want to use this engine to teach people to become what we call 360-degree scientists. So the PhD students shall really, so the PhD student that works in an informatics lab will not just do informatics. He or she will also work in the greenhouse and also do the biochemistry, the whole mass spectrometry. So that we actually train people, the new generation of plant proteomics specialists that have seen the whole thing. So the crop, the protein extraction, the mass spec, but also the bioinformatics part. And we actually combine this also with a lot of different soft skills and many possibilities to really sharpen their skills also outside of the daily work in the lab or in the greenhouse, right? So intercultural competence, teamwork, all this stuff. So we really want to try to get to make the best plant proteomics specialists you can imagine. And so yeah, one more time, if you have interested and talented people who can imagine to do their PhD together with us in Germany, it's a great country, and Germans can actually also be quite funny, something people do not believe. Let us know. And with this, I think we're also at the end. So I think we want to thank the whole team. I especially would also like to thank Zara, who actually works with us on this crop proteomics project. And she actually did most of the wet lab preparations parts of us. So she established the workflows that we now use for the whole project. And yeah, thank you for your attention.
Thanks a lot, Prof. Bernard Costa and Dr. Nils Rogen for this wonderful eye-opening speech. This is, to be honest, inspiring to all of us who's doing proteomics or planning to do proteomic research in the currently or in the near future. I'm sure the audience have questions, and we still have about 8 minutes until Prof. Sebastian joining us. So I think I would like to open the floor for questions from the audience until we get our last speaker to start his talk.
Hello.
Yes.
Hi, this is Aizat. So I'm just going to start it off. First of all, thank you very much, Prof. Bernhard, as well as Dr. Nils, for the enlightening talks. Basically, we are at the Malaysia. We have a society, the Malaysian Proteomics Society, which basically give a lot of values in terms of proteomics research to our countrymen. Anyway, in terms of, I've got a couple of questions regarding the, perhaps Prof. Bernhard first. Do you have any type of mass spec that you use in your study that will be of useful to, in terms of getting more proteomes? Because you said about the dynamic range and everything, trying to compare between transcriptome and proteome. Is there any type of mass spec that you use in your study?
Yeah, we basically collected the data also on a single mass spec platform. In our case, it was an Orbitrap HS. But I think today, with a modern high-end mass spectrometer, either from Thermo or from Bruker, it really doesn't matter so much anymore. So it's not that in terms of the mass spec performance, they are quite comparable. It's more the overall workflow that determines how deep you can go and the amount of time you're willing to spend on a given sample. So the more upstream chromatography you do, the deeper you can go, but that also means you have to start with some more material, and it also may mean that you have to spend more mass spec time. So I think on the whole, what we have done is nothing extraordinary in that sense. We did some upstream chromatography separation over the peptides and then used a high-end mass spectrometer to read it out.
All right, awesome.
But clearly, the more current the mass spectrometer is, the more sensitive it usually is. But right now, I think it would be a very difficult choice whether you should use a Bruker timsTOF or an Orbitrap Fusion Lumos or an HFX or an Explores. They are all very high performance instruments.
Okay, so basically in your lab, basically you utilize any type of in Orbitraps, timsTOFs, and everything in terms of the
Well, we don't have a timsTOF, but as I said, I don't think it matters much these days. It's more the overall package from sample preparation to the informatics that determines how deep you can go.
All right, awesome. Thank you, Prof. Perhaps I have got another question for Dr. Nils. When you asked about the different types of crops and plants that we are interested in, do you have any sort of what we call it, plants or anything that of interest to for the profiling? So I'm not sure whether are you interested in a tropical kind of plants, or is it going to be fruits? Any type of plants that you are looking for?
Well, we have a huge list with a lot of gaps to fill, but it's everything. So it's basically each and every crop you can imagine.
Right.
It's really so a list of 100 covers, I think, the most important things that you and I know. And we really want to, so we do not just want the fruit, we also then want the leaves, the roots, the seeds, etc., the pollen.
Right. So this is for the proteomes, isn't it? So the proteomes of the plants. Okay. Right. Cool. So are you looking into like the other omics as well, or just proteomics?
This time, I think we decided to focus on proteomics. We have, I think, some ideas for metabolomics projects, but we do not plan to do metabolomics for all 100 this time.
All right.
Also not transcriptomics.
So the list of the 100 is somewhere in the website?
Not yet. So the website, I think, Bernard, is still under construction, I fear.
Well, we're basically just gotten that funding, so we have a URL, but no content yet. But that's a good suggestion. We can actually put the list on the website and also mark which ones we already cover by some of the partners, and then our future potential partners can actually just take a look whether there are any plans on there for which we don't yet have partners.
Awesome.
So I think what I could suggest is that I send the most recent version of the list to Jamil, and Jamil, could you then forward it to people who are interested?
Yeah, sure, definitely. That will be a nice idea.
Yep. Just one last thing. The students vacancy is only for PhD student, not for postdoc or any postgraduate. No, it's for postgraduate, but not for postdoc position, isn't it?
Correct. So this is a specific funding scheme from the Bavarian government to invest into PhD students. Right. And we're going to run a very competitive selection process where we advertise this worldwide, and the selection process really goes in 3 steps with the idea in mind. The German funding agency really pushes us to only recruit the best people we can find. So it really has that mission to try and get the best from wherever we can get them, and that's the international part. And the selection will go. I mean, we're going to ask people to apply and send their CVs, and then we're going to just go through the CVs and make some preliminary collection, and then we're going to set up a lot of Zoom interviews with these people. And after the Zoom interview, we're going to invite 20 candidates for a whole week to Germany to be on campus with us and see all the PIs, get to know the project, get to know the Munich area, get to know our university, and then from those 20, pick 12 to whom we're going to make an offer. So that's roughly how it's going to go. But still, we don't want to discourage anyone to apply, but this is also why we're using this forum here by encouraging people. Great talent is available all around the world, and we want them to know about it. And if they're interested, we want them obviously to apply, but then they of course need to also know that it will be a competitive selection procedure. But again, when I was a PhD student, I also applied to a very competitive scheme, at least I was told, and it was for women mostly, and it was to only spend a bit of the time abroad during the PhD, and I ended up getting it, even though I spent the whole 3 years in Oxford. So we never want to discourage anyone.
Right.
But it's important. But it's important to find what we think, or what you think are the best people.
Yep, sure, we do. By the way, can perhaps the links will be given to Jamil later on. Perhaps we can actually give this out to promote this out to our members. Perhaps there are some qualified members out there, and perhaps they want or they are interested with this position. Thank you very much, Prof, as well as Dr. Niels. We'll continue on. Back to you, Jamil. Thank you.
Thank you, Dr. Isaac, and of course, thanks to Prof. Bernard and Dr. Niels. I think this is a very great opportunity for the best of the best people who is interested in doing PhD in proteomics. I wish I have had this chance maybe some 15 years ago, but I think it's too late. So I think attachment, as Prof clarified, is just for PhD. It's for the best of the best candidates for PhD students provided. So I think we have few other questions in the chat box. I will leave it to the end of this webinar because I think we have already Prof. Sebastian already joined us. He's already here. So good morning, Prof. Sebastian. We are moving now to the second part of the webinar. So let's all welcome another distinguished speaker, proudly a friend of mine and all the friend of mine, Professor Dr. Sebastian Carpentier. Prof. Sebastian Carpentier is a bioscience engineer specialized in agriculture and gene biotechnology. His team focused on phenotyping the biodiversity of crops by integrating a biotic stress physiology at plant and tissue level with omics technology at cellular level. In the framework of international mobility, he was awarded a postdoc fellowship by the Flemish agency FWO at Wageningen University in the Netherlands. Before joining the consultative group of international agricultural research as a team leader for the banana characterization and evaluation, he was a professor in crop stress physiology for 6 years at the Catholic University of Leuven. He is managing currently the KU Leuven Mass Spectrometry Facility, CBioma, since 2011, by characterizing the genomes, evaluating the traits of genotype preserved in the gene bank in the high throughput, and by validating their suitability for certain crucial agroecozone on field around the world. His research is crucial for feeding pre-breeding programs and distributing the crop's gene pool. Without further ado, let's have all Prof. Sebastian Carpentier on board. Prof. Sebastian, the mic is yours.
Hello, good morning, everybody. Do you hear me?
Yes, Prof, we do.
All right.
I cannot see you yet.
Hear me?
Yes, now, yes.
All right. Excellent. So it's strange days nowadays. We work from home. But luckily we have technology to get our knowledge, right? The idea that I'm going to start my presentation?
Yes, Prof. Sebastian, you can present your slides.
I'm still struggling with yeah, that's the one. Oh, no.
All right.
My presentation, yes. Okay, let's try to get it in presentation mode.
Yes, now we can see.
Excellent. All right. So, good morning, everybody. Indeed, thanks for the kind introduction, Jamil. Sorry for being late. It was quite challenging for me to get here on time, so thank you for adjusting the schedule if necessary. For the next half an hour, indeed, I will give you a brief overview of what we do here in Leuven. Indeed, it's specialized around crops, and we try to integrate different technologies because I believe that's the way to go forward. So that's a classical slide. Probably you have already seen it in many presentations, but I bring it back because it's really quite important. As you know, transcriptomics and proteomics, they are really showing what at this moment is happening. So the kinetics are most important, and you tend to miss a lot of interesting kinetics if you don't measure. This is why I would like to stress to phenotyping, because there you discover a lot of interesting moments to take your samples. We all know transcriptomics, proteomics, metabolomics, those analyses are still expensive. And so it would be really not wise to take the samples on the wrong moment. So that's why I would invite everybody, please try to learn as much as possible about your samples, about your individuals, and think about the right moment to take your samples, because then you might get the right times to take them. Of course, you know that both omics technologies, or the three omics technologies, are for sure complementary. timing-wise, kinetic-wise, of course, the transcriptome is ahead of the proteome, so you should realize that. And not always there are, of course, are correlations. But the most important is to look towards the dynamics. And of course, we talk about crops. A lot of crops are still not sequenced. Great help if you have let's say a reference genome or something that this sequencer is very close by, because then you can start doing the transcriptomics de novo assembled, and you can search your proteomes, your spectra against your transcriptome library, which is in my eyes a very important approach. But of course, we are talking about crops. There is a lot of challenges if you work with non-model organisms. The first one was, of course, sample preparation, protein extraction. Some tissues like fruits, like other things are quite challenging. Yes?
Sebastian, I'm sorry. I don't know. Maybe it's only for me, but your slides are not advancing. I don't know if everyone else sees them advancing, but you're not in presenter mode.
Okay. Good to know. For me, they are advancing. Should I stop sharing or?
Prof. Sebastian, I think you may off the webcam because of the maybe connection, and try to present the slide again. Then we hopefully they will be moving.
Yeah, and what do you see now? Still the first slide, or because I share my screen, so you should now see yourself, Jamil. No, right?
Yeah, we don't see anything currently.
Okay, let's try again. Select window or screen. I will try to do screen 1. Allow. So what are you seeing now?
Yeah, now we can see your third slide.
And is it in the presenter mode or is it full slides?
It's not in the full mode yet.
Okay, now it should be, is it?
Yes.
All right.
Maybe you want to go back one slide, go back one slide, go front one slide. Yeah,
exactly.
It is moving. Yeah. Fantastic.
This one you didn't see, right?
Just now, not really.
Okay. So as I'm explaining, you have the different levels of the omics technology, and I was saying the pheno, the first one is the very important one because of the dynamics. It will give you really indications of when to take your samples. And I was saying that although of course the core facility of my business is proteomics, I would really be an advocate for doing transcriptomics, because especially if you're specific, let's say cultivars that are not sequenced, it might help you a lot, and it might give you the right alleles, as I will demonstrate further on. And then I was up to the challenges. Of course, if you work with non-model crops, tropical crops, whatever you have, protein extraction can be definitely an issue. A lot of interfering compounds might be in there, polyphenols, name it. It might be a challenge. Luckily, of course, a long time ago, we found a solution for that. It's a matter of really paying attention to the sample prep. And I fully agree with what Bernard said before. The type of mass spec is nowadays, they are all quite well advanced. Is less important. Don't think I will buy a mass spec of 800,000 that it will solve your problem. No, we pay attention to the sample prep. It really needs to be good from the beginning and take some time to optimize everything. The protocols needs to be optimized according to tissue, according to species for sure. And the second issue might be that, of course, your preferred variety is not sequenced. Then we have some solutions. A long time ago, we published this. You can work with spectral library searches, spectral clustering, and so on and so on. But if you have some budget, I would really advocate to go for mRNA-seq, or of course, not against a reference genome or towards a close relative. And then search against those assembled contexts. That really helps forward, as I will demonstrate later. An additional issue might be that some crops are diploid, so it's the mixture of genomes. We all know a well-known example of wheat, which is hexaploid and contains different genomes. For instance, cotton is also such one. And Jamil introduced me as working on mainly on bananas, and bananas are also polyploid species, as you will see. So that's the polyploidy and the bringing in of different genomes and specific alleles makes it really interesting and can make the difference in the phenotype. But it's really crucial to hunt those down. So let me get with this case. We published this a while ago, where we worked on the integration of transcriptomics and proteomics on bananas, on those polyploid crops, and where we really tracked down, hunted down those homolog expression and get some clues why specific genomes bring in specific traits. As I said, it really begins with characterizing. So here you see 3 triploid bananas. One has one A genome and 2 B genome portions. The other one contain only full A genome portions. And as you see, the root growth of this ABB is considerably even under control conditions, so no osmotic stress. And under osmotic stress, you see that the root growth continues to grow, which is an extremely interesting characteristic. And then, of course, we are tempted to assume traits come from the B genome, right? This is tempting, and that's why we started to see what is differently expressed in this ABB triploids than in the other two. So we combined transcriptomics and proteomics, and as you can see here, yes, indeed, the transcriptome is significantly different between the ABB variety, which is the orange one on the left, and the 2AA variety, so the green one and the red one. So it's a big separation. You see a complete difference, well, not a complete difference, but there's a significant difference that you can say that those transcripts are correlated towards the ABB genotype. And so this is the separation in the first component. And the third component is, of course, the control treatments from the stress treatment. So that enables you to really point down transcripts that are really up or down regulated and only reacted, for instance, here on those 2 AAA varieties and not in the ABB. You can track them down on the chromosomes where they are. Breeding, the proximity of genes is quite important because 2 genes together to be inherited together, right? The same is true for this ABB. You can really pinpoint transcripts that specifically react to the same conditions of the ABB variety, and then you can start thinking about this, seeing what families are they, what are they doing, and so on and so on. Of course, the proteomics, again, here I must say, please pay attention to sample preparation. Please pay attention to your chromatography. So you see here a separation on a long column, a 50 centimeter column, and separation during 200 minutes. It's really important that everything is well done, that everything is well separated. And in terms of identification and speed of the mass spec, that the number of ions at the same time is kept under control, because that's still an issue, as you will see in the next slide. So here you see the relation between the abundance of an ion that has been quantified in the mass spec and how many molecules, let's say ions, occurred in this abundance. So basically, you see a nicely normally distributed pattern of peptide abundances. So that's what we could track down quite as you can see. In red, you see that's the ones that were selected for an SMS. As you know, selection for an SMS really depends on the abundancy of your parent ion, and if it's not abundant enough, it will for sure give you a bad spectrum. So we all program our spectra or our specs in the blind approach, at least, to focus on the most abundant ions, and that's why you get this bias, of course. So the low abundant ones are simply ignored. So in red you see indeed the bias shifting towards the more abundant, towards the right, and then of course get a successful ID, you even get a more shift to the right, because as I said, if the mother ion is not abundant enough, it will really give you a poor quality spectrum. That's obvious. So the pinpoint is the orders of magnitude. So abundances are what they are. What can we see? Okay, let's ignore these abundances. Let's say, okay, let's count from here, 1, 2, 3, 4, 5, 6. Let's be optimistic, let's say 6 orders of magnitudes of dynamic ranges we can see in the proteomics, which we can only identify, of course, the most abundant, which is obvious. If you have a look at the transcriptomics, because this week we took from exactly the same plants, right? And you immediately see dynamic range is much less. You should definitely ignore reads below 10. And so you end up with 3, if you're optimistic, 4 orders of magnitudes. So there you already see the complementarity between both. You see much less, but can you have a high resolution in discovering differential abundances, right? While with transcriptomics, you see basically everything that is expressed. If you have a really deep sequencing, you see really everything that is expressed. But the differences need to be bigger to call them significant. That's a fact. So if you then check towards complementarity and you apply strict statistics in terms of what is significantly different and interesting for you, you see, let's say that you, of course, with mRNA-seq, because of the numbers, you pick up much more. While that the correlation between transcriptomics and proteomics is not one-on-one, and minority is confirmed in both technologies. So that's why, for sure, they are complementary. In terms of success rate, you see basically that it's the same. 12% of the proteins that we could quantify was significant in proteomics, while 10% was in mRNA-seq. That's also nicely illustrated in the next slide here. And you see abundant reads. So you see here the read counts of transcriptomics, and you see you should discard what has low reads, because you simply, the sample size is too low, so you cannot trust classification. And then you see, of course, you have I think you go much deeper, but then of course the proteomics basically exactly the same. So you basically covered the abundant transcripts, and a useful exercise for later on, if you say, let's go proteomics, to have an idea where you can go, and is it realistic to pick up low abundant expressed genes, of course. But let's jump into the proteomics, because one additional advantage of the proteome is that part of let's say blind mRNA-seq, that you can really take allele specific proteins without any prior knowledge. What you see here, you see the separation, the blind separation of the proteome, and you see indeed you have proteins for the ABVs on control and stress conditions, and the same is true for the fully AAA varieties. When I say you don't need any prior knowledge, you see here that are only detected in ABV plants. So it's really tempting to say that this is a protein that is encoded from the B genome. It's completely absent from the A genome, and without any knowledge, even without being sequenced, you can see that, because you quantify a parent ion that is only present in one type of plant. So that, of course, it needs to be further validated. That's what we came up purely by quantifying let's say the label-free quantification of the parent ions. We could really pinpoint out peptides that were unique for the AAA genotypes and that were unique for the ABB genotypes. And now, of course, I hear you thinking, oh, but this ABB also has. Not expect that some proteins are quite uniquely present on the AAA. Correct, you would expect them, let's say, to be present in a ratio 1/3, 2/3, right? Yeah, but we discovered to sequence this ABB variety, and it turned out to be that it's more complex than that. You have chimeric chromosomes that, and some are like chimeric parts has been replaced by a B. So we discovered in our collection that not all ABBs are the same. The proportion of A and B genomes is not simply 1/3, 2/3, but it really depends from individual to individual, and that's really interesting, but also, of course, also at the same time quite complex. Of course, as I said, against our mRNA-seq, because then you make a library of your specific genotype. And of course, you come up with causative SNPs. The reason why a peptide is unique because the sequence is not the same, and that's what is illustrated here. So you see both spectra, everything down Y10 is exactly the same sequence, and this, of course, we discovered in our mRNA-seq sequences that we have a Q here and an H there. And of course, it makes sure that this peptide is only detected in an individual B genome, or at least containing this allele, while of course it's not present in full AAAs. Some other examples. Sometimes we could not find SNPs because the sequence of the peptide was exactly 3 libraries. But we did see, if we looked at the quantification and the read counts, that indeed it was much more abundant in EB. So there we suspect we have SNPs in the promoter regions, or there might be epigenetics, let's say, on this specific chromosome that it makes not accessible for transcribing. So that's why the expression is quite low. So that's why, of course, it's always interesting to see. And then in some cases, the sequence was exactly also not the same, but we could not see any difference in quantification of mRNA-seq. And that's a nice example where indeed there is no correlation between transcript and proteome abundance. And of course, to think about protein half-lives of as I said, the kinetics of transcriptomics and proteomics are not the same. Once you have all your interesting, let's say, alleles, you could try to do a gene ontology enrichment and see to which classes they are connected, and then you can build your story. Of course, for sake of time, I will not go into that. But then, of course, you plot that on the physical maps of the chromosomes, and this is really interesting. So that was the story of, let's say, the throughput discovery. No extensive kinetics, no specific zooming in on cells, because I want to build up the story coming from time throughput to, let's hope, one day into single cell proteomics, right? Let's see when we get there. But step by step, we took several things. Just in between, was this new that we could pick up those allele-specific proteins? No, because in 2011, we were already doing the same with 2D gels. Of course, is the rate when you discover the throughput of the B gels is just absolutely zero. You just stuck to the 2000 spots you're on there, and so it doesn't work. But at that time, it was sufficient to blindly also cluster all the different genotypes, which was quite interesting and which gave us the ideas to scale this up. Of course, am I saying that the gels are completely to be forgotten? In theory, yes, but there are some exceptions that you still need them. And on the left side, this is one of the examples. If you're interested in protein families, like the heat family is in every species a quite complex family of a lot of paralogs. If you then combine this with the complexity of an polyploid, then you create much more redundancy. And if you want to follow them all, 2D gels are really still interesting, because you can see the spots of the different isoforms, and if you pick such a spot, even within the spot with a mass spec, you will see that different isoforms are in there, and then you can start to quantify. So there you can make the resolution to see that all, and of course to pick up also the lower abundant ones. Of course, once you know the prototypic peptides, you could go back to a gel-free approach, but I don't think you will discover them all if you start from a gel-free approach. So that's again, let's say, please don't forget to integrate omics technologies. And then let's jump from the bananas towards other crops. This was a collaboration with some German colleagues where they were working on rye, and they discovered in rye that some rye individuals have an additional small chromosome. They call it a B chromosome. And this indeed can be inherited from generations on, but the copy number also vary. And so they were interested, knowing the story that we had technology to pick up those, let's say, specific proteins. They were interested, of course, to hunt down the genes that are only encoded into these small B chromosomes. And you see here an example. It's really, without any previous knowledge, if the peptide is unique, you really pick it up. If you want to know more about how to start also this allele-specific approaches, this has been published in a book. Approaches, whether you have access to mRNA-seq or to go further reading there. And I would like to spend the last of my presentation here on the dynamics. You'll see here an example of if you don't know what is happening to your plants, you will simply not know it. What we've done here is that we stressed maize plants with sodium chloride, and you know, if you feed sodium chloride into the roots, let's say 15 to 20 minutes later, the ions arrive in the leaves, and something nicely happens with the pH of the apoplast. It changes, and that's what you see here, right? This is the time point, and you see bit later, it's around 15 minutes, that you see the pH changing and then going back to normal. Of course, if you take a sample here and you take a sample there, it's changed. Then, of course, you missed a really important time point. And that's what we basically did. We took several samples along the time. And, of course, we wanted to know how is the proteome changing towards a change in apoplastic pH? Does it do something? And the answer is yes, of course. We had the necessary controls when we added the ions with a buffer so that the pH could not change. And that's basically what you see here. Let me go back. The pH is really quantitative. If you add more salt, so if the concentration is higher, you get a higher weight, and that's what you will see in the movies. So you see the pH change because you have a dark color depending on the pH, and then it gets to normal again. And you see more the wave was earlier, then we added less salt. So that's really to convince you, pay attention, take the samples on the right moment. And exactly as you see, proteome of the samples that were taken where the pH could change in those two conditions, we could really pinpoint that only reacted towards a change in apoplastic pH, and we could deduce everything that was related to the stiffening of the cell. Really interesting. To end, because I promised that we would go to the single cell. It's not single cell yet, as of course everybody knows, right? Everybody is still working towards that. But because we go towards isolating not a tissue anymore, but single cell types, and that's a story of guard cells. So the guard cells are part of the stomates that go open and close to regulate photosynthesis and water loss, as you might know. And they are very dynamic. They react to light, they react to drying of air, and so on. And so we wanted to see, is it possible to study the guard cell proteome, and especially in some interesting dynamics? And the answer was yes. So this is here a proof of concept in Arabidopsis. And what we did is we took samples 1 minute before the light was on, 15 minutes after the light was open, and then 1 hour after the light was open, and you can see all proteomes are completely different, and that covers that indeed in open stomates, we cost a lot of energy because a lot of ions need to be pumped, not taken from the light because everything is already present 1 minute before diurnal regulation, and the proteins are ready to react and deliver the energy as soon as the light goes on, and the energy comes from fatty acid oxidations, so not from the light, not from something else. So that was really interesting to see that if you combine specific cell types and specific dynamics, you really discover interesting things. The step further is, of course, because the previous one was still, let's say, the abundant proteins, but once you know what you're looking for, we know that abscisic acid receptors are quite important in guard cells, and of course, if you work with all is sequenced, all is known, and you can proteomics, the expression of the specific isoforms, and here in this paper, we could really see both combination of transcriptomics and proteomics of what the individual, let's say, paralogs are doing, and what is their role in stomates towards reacting to what conditions, and how the integration is done when the signal is given for the stomates to close. To end, this is an example of guard cells in my bananas. I went back to my bananas, and we took, again, looked towards dynamics. We played around with different NPDs, so different dryness of the air. Of course, if the air gets drier and hotter, stomates will close and transpiration is reduced. And it's in excellent correlation towards how much a certain individual was transpiring at that moment and its specific proteome. So this study is not finished. We are fully working on that. I hope that meanwhile I convinced you that of different omics and especially getting into the right moment is really super interesting and will bring up new things. Thanks for listening. Of course, this work has not been done alone. Cooperation on it with lots of funding, and that's my team a while ago. And I would like to thank you for your attention. And if you have questions, reach me on both email addresses. Email.
All right. Thanks a lot, Prof. Sebastian, for the great talk that stressed a lot of the importance of sample preparation before starting any proteome work. So I think I would like to open the floor for direct questions to any of the respected speakers. We have also a few questions from the chat box. Maybe we can start to answer them. There's a question about the process rescoring and how it was performed. This is to Dr. Nils, if you want to elaborate on that.
Yes, but I'm not sure if I understand the question. So do you mean how project works in detail, or what we did with our data?
Maybe Dr. Go want to ask directly the question to make it more clear. Go. I'm not sure if Go getting me anyway. Maybe until he.
Or maybe I can jump in. I mean, a high-level answer would be the way that PROSIT works is you allow a search engine, MaxQuant or another one, to not only return the best match peptide to a spectrum, but anything it can match it to, regardless of how good the score is. And then PROSIT will reevaluate those hits in light of the intensity distribution of fragment ions. And then sometimes that happens that the second hit is promoted to the first, or something that really was a hit that fell under the score threshold of the search engine that gets promoted up, or sometimes it switches the order. The second best hit by the search engine is actually what fits better to the fragment ion intensity distribution. So that's roughly how the PROSIT rescoring works. I'm not sure if that answers the questions, but that is the long and short of it.
All right, thanks a lot, Prof. I think we have partially tackled this question about the plan for doing other omics besides the proteomics for the crop atlas in the future.
Yeah, so maybe just so we are not going to do that because we simply don't have the resources to do transcriptomes and metabolomes on all of them. But actually, a lot of our partners tell us that they have at least partial genomes or partial transcriptomes, and we're going to, of course, take that data from a point of view that, to what Sebastian was saying, if you don't have a reference genome, the next best thing you can use is then a transcriptome. And in fact, I mean, we are super happy that we have the EBI, the European Bioinformatics Institute, on board. They have just launched a very strong biodiversity program, or in the process of launching that, where they want to genome sequence a lot of also plants this time. And so we're working with Wolf Abweiler and Paul, now what's his last name? Anyway, so we have strong ties to the EBI, and we're actually going to send many of the students that we're looking for also to the EBI for 3 months with the view to help annotating these genomes using proteomic data. So by all means, we need that data, but we haven't got the resources to create them ourselves, but a lot of our partners have.
All right, excellent. Are there any questions from the audience to Prof. Bernard or Prof. Sebastian or Dr. Nils? Actually, until we get a question, myself, I got a question. Just now, Prof. Sebastian stressed the importance of sample preparation, and earlier, Prof. Bernard did mention something personally surprised me, because he did mention that Robisco could be in the leaf less than 10%, but some papers claim the content up to 65%. So is there anything, Prof, to do with the sample preparation or the way that the proteome has been done? Because it's quite different between less than 10% and 65%.
Yeah, so that was one of the major surprises. In part, we think these numbers came up a long time ago and created by classical biochemistry, and sometimes checking preparations even on protein gels, and then you see a big blob of Robisco somewhere in the gel. But if you then use a mass spectrometer, you find 500 proteins hiding behind this big blob of what, of course, is mostly Robisco. But I think the biochemical methods that were used, let's say in the 60s, when some of these numbers came up, were just so not as sophisticated. So and that's where easily these estimates could come in, because a very clean way of doing it would have to have a very clean prep of Robisco, let's say, and do amino acid analysis and count the moles of all of the amino acids and relate that to the total. But that was one of the big surprises, actually. And don't quote me whether it is less than 10% or about 10%, but the difference to what you were saying actually was quite large, surprisingly large.
Yeah, that's, I mean, regardless of it's at 10% or even more or less, but it's surprising compared to what we used to read about the leaf specifically.
Correct. I mean, I'm a trained biochemist too, so I've been trained by using Streyer's textbooks and Albert's textbooks, and it's surprising that 20 years down the road, when you take another look at some of those global statements, you really have to question that occasionally. So there is more protein expression diversity in those leaves than what people thought. But the same goes, let's say, also for antibodies and how specific they are. We know that most antibodies are not very specific, but everybody teaches their students, antibodies are the most specific protein detection system you can imagine. Also no longer true. I mean, one occasionally has to question the old truths and adjust them a little.
Yeah, correct. Well, I think this is, not to take home for us who's doing proteomics, especially if the sample was the leaf, not to really stick to those old statements and maybe dig deeper to look for more proteins rather than the Robisco, which claim to be like half of the leaf. If there's any question from the audience, we would like to have, if there's any. Well, until we get another question, maybe another question from myself, if Prof. Sebastian and Dr. Nils do not mind. Earlier in both speech, there were a highlight about the sample, and those samples, including the pollen, including the seed, definitely very important to get enough amount of protein, but it's very difficult at the same time. So is there any advice on how to compensate this difficulty with the, how to say, I mean, there is not enough maybe material, but still there is a certain amount of protein or a certain concentration, to be specific, need to be met before we proceed next. So for those who is interested in joining the Proteome Atlas in the future, or doing their own research on samples, specifically with this kind of difficult tissue, is there any kind of advice or any direction on where we want to go about that? Maybe Nils want to start, and maybe can have Prof to comment on that.
Sorry, I jump in before Nils can give a better answer. But in a naive way of thinking, and I sometimes choose to think that way, if we're talking about the 100 most abundant crop plants, material cannot be an argument. If millions of tons of anything is being harvested every year, it's to me sounds more like getting it organized. I can see that this is a whole different difficulty, and I know that some plants only flower once in 20 years, and that is clearly a problem. But still, when thinking about millions of tons of crop every year, there's got to be some palm tree that is flowering some on some part on this planet almost every day. So it is maybe an organizational matter. So we hope that we can overcome these problems by encouraging our partners to really try.
Yeah, exactly.
But then at the same time, we have decided that we're going to work with anything that's practical. So let's say if for a certain tissue, like in pollen, if we can only get 20 instead of the 200 micrograms, we know we can get a fairly decent proteome from 20 micrograms. So it's not a showstopper. Given that also the timelines along which many of those plants either grow or being harvested, we try to get it right in the first submission of sample rather than in the third. But it's not a showstopper. So we'll take a practical approach to it. If our partners tell us, look, we've done everything we can on collecting pollen and it's only 10 or 15 or 20 micron, we'll happily take it.
All right. Okay, that's good news. That's good. So organizing is really matter. Any comment from Prof. Sebastian or Nels on that?
Go ahead, because you were cut it, right?
You can just go first. I guess everything has been said already.
No, I agree what Dan had said. I mean, it's the same with our guard cells. Guard cells are really not abundant. Even if you see that we managed to get a really decent proteome out of Arabidopsis, which is a tiny plant which has tiny leaves. It's a matter indeed of enriching and having the right sample prep. So that I fully agree is not an issue anymore. It depends on what you also look towards the single cell proteome. If you say single cell proteome is from a single cell type, then it's absolutely not a problem. If you say single cell proteomics from one cell, a completely other story, right? We are far from being there, and again, I think the method is there from the sample prep, and people are working hard on that. Also from the school side, we know that for transcriptomics, people are already doing that, from one cell a transcriptome. But of course, we know that indeed mRNAs you can multiply by PCR technology. For proteins, it's not the case. But I guess being more sensitive, and we will get there. The plant world will, of course, lag behind as always. But yeah, platform is already a huge leap forward, and that's the way to go. Otherwise, proteomes are really mixed. In the past, people tended to also do this Malbitops where they could do imaging, and then you could see also the relationship between the place. Also, this will get better and better, I expect. So step by step, we will get there. That's also my belief. How long it will take, that's another question, of course. But for me, it's important to know the different cell types, so for sure they have a different expression pattern.
All right, great. Nils, anything to say about that? All right. So yeah, I think I need to remind our participant that there is a link for there to register and for certificate. Is there any question from the audience? All right. It seems that we have no more questions from the audience. So before we end up, I would like to invite all participants for a photo session, if you guys do not mind. So just open your webcam, and yeah, this is the pandemic style. So just open the webcam, and I have to count 1, 2, 3. Hopefully, Dr. Nurul can can catch us all on the camera. And yeah, hopefully all the rest of the participants to, as much as you can, open your webcam, and maybe we go for photo session for this webinar. All right. The cameras, please. All right. More cameras to be on, hopefully. Okay. All right. I think we can now take 1, 2, 3. All right. Maybe another one. More cameras if possible. All right. 1, 2, 3. All right. Can we get a thumbs up from Dr. Nurul that we have gotten some pictures? Are we done?
Yes, sir. Yes, already done.
All right. Okay, that's great. Fantastic. All right. So I really would like to thank everybody for being with us. And at the end of the webinar, I would like to highlight that this webinar could be a good starting point to invite all to collaborate, whether directly to collaborate with the ambitious project, Proteome of the Crops That Feed the World, or collaborate with us here in Sultan Idris Education University. I think this collaboration, when it happens, will come out with a high quality research and hopefully high quality outcome. Finally, I would like to give a very, very special thank to our distinguished speakers, Professor Dr. Bernard Huster, Professor Dr. Sebastian Carpentier, and Dr. Nels Rogen for very kind willingness to share this interesting latest update on the proteomic research. Really appreciate that and looking forward for future collaboration with all of you. Dear all, sadly, for me, this is the end of today's webinar. So thank you all for being with us today, and hopefully we will see you in the upcoming event. Have all a great day. Thank you so much. Salam.
Thank you very much, Prof. Bye.
Bye-bye.
Bye-bye.